I have to start by apologizing that this is a very strange type of kind of lecture, the hippocampus as a topic in a way. And also, it's extremely difficult to judge what you know about it and what's not. You are a diverse group, so some of you might find it completely boring, especially the talk of today, which is in a way more like textbook talk and a bit boring, but maybe it will just save you a lot of reading. And mainly what I was trying to do is organizing kind of probably many things that you've heard of in different ways, but were not like in a context. So just you have to admire this amazing picture of the rainbow. You all know about the rainbow, right? Anyone never heard of the rainbow? No? No? Okay. So I'll just tell you one thing. The idea is that you kind of you express proteins that are fluorescent with different combinations, random combinations of the protein. So if you kind of then illuminate it with different light, excitation lights, you can actually differentiate between it. You can see it on this resolution. It's really pixelated, I can see that thing. But just Google it later and you can see that basically you can trace individual neurons one next to the other. And the original idea, which was very nice, was that you would be able to do tracing of networks using that, follow a single axon and kind of see where it connects to different neurons. It wasn't that successful in that sense because the optical resolution is not good enough, but it's just making beautiful pictures. Second thing I wanted to tell you, which is kind of strange, is that you probably have heard that Lou Reed has died 2 days ago, and I thought it would be interesting to mention it here, important to mention it here, because you are at the beginning of your PhD and you are all kind of excited and very happy. But if no one has told you that before, PhD has very, very profound periods of depression. When things don't work and your peers are already going to postdoc and you haven't published anything. And at those times, he will be your best friend. And believe me that it doesn't matter how deep you go, he'll always kind of, be on the one step lower, smiling at you. You haven't found it to the bottom. So anyway, so what do you know about the hippocampus? Do you want to have? Yeah.
Can you raise the microphone?
Really? I thought you kind of recording. Do you hear me now? Is it better? Okay. Which recording? The video? I don't know if they're taking it. Do they?
Yeah.
Really? Oh my God. Okay. No, because I was looking for some historical recording and lots of them are missing. So it's okay. So what can you tell me about the hippocampus? What do you know about the hippocampus? Just throw kind of words, CA1, yeah. What? Consolidation, yeah. Place cells, yeah. Shape and collaterals, right? What? Right. Okay. So yeah, so it's kind of it's a mixture of things. Just before I continue, I wanted to mention this book. This is a book that was written in 2007 by a group of actually people that have been working on the hippocampus for a long time. Per Anderson and Tim Bliss were the first people to find. It was Tim Bliss, but he was a postdoc in Per Anderson's lab, the LTP. No one actually mentioned LTP. Richard Morris is a famous guy. He developed the voice water maze. I think David Amrei is an anatomist of the hippocampus, and John O'Keefe was the first one to record the play cells. So this is a very good book. It kind of covers everything that you can think of. It's almost up to date, 2007. It's pretty good. So if you need a reference for the hippocampus, that's a good one. Okay, so I think that one of the things that I realized preparing this lecture is that the hippocampus comes up in kind of two major contexts, and they're kind of surprising because whenever you go to a scientific meeting, a neuroscience meeting, you'll at some point or another you'll encounter the hippocampus, but a lot of that is just because it's a good model system, not so much about the hippocampus itself. It just happened to be like the dream system that you can think of in many for many scientific questions. And then what you'll find out is that you'll talk with people and they will tell you, we're studying memory, but basically what they're studying is synaptic physiology, and this is what and they just happen to do it in the hippocampus, and they don't know anything about memory, and they don't know anything about learning, and they don't even know what the input and outputs of the hippocampus are. They just kind of know how to slice the hippocampus and work on it. So we'll actually concentrate on that today, and the other one is a good model system this is a good model system in synaptic physiology, but it's also a good model system for spatial navigation, for example, which is another property, another thing that was found in the hippocampus. So this is another case where it is a good model system. By kind of accident, it happened to become people realize that it's very important for memory, and then there's a whole different class of a different aspect of the hippocampus. So I think like you would hear about it, under this context a lot. And then the rest is more like under, just because it's a good model system. The issue of disease is a different one. I don't know how many of you will ever have to work, to deal with that or interested in that, but hippocampus plays a huge role in epilepsy, and also in Alzheimer because it's obviously related to memory. So, but we'll deal with that at the end of the course. Okay, so what is the hippocampus? So the hippocampus is just part of the brain. I don't know why it does more. Okay, it's a part of the brain. It's located under the temporal lobe, kind of within in the human, kind of wrapped within the temporal lobe. The auditory cortex, the primary auditory cortex is around here, and it's obviously on both sides of the brain, and it's pretty isolated system in the sense that you can take it out, right? It's in its entirety, whatever you say. So you can just, if you do dissections, you can really just take the whole hippocampus out, and obviously because of the way it looks, it was given the name hippocampus, which is a seahorse. The original name is actually, it doesn't look like that at all. The original hippocampus is a mythological animal, which is a fish and a horse together. But anyway, in the human, it's considered part of the limbic system that we will talk about when we'll talk about emotions. which is also the amygdala and the other parts of the system, which is kind of surprising in some sense, because in some ways it looks more like cortex than any other part of the limbic systems, which are, just nuclei, which doesn't have almost any order in them. The hippocampus is very organized, and it's also kind of very intimately connected to the cortex, to the neocortex. So it's kind of surprising, but nevertheless, it's part of the limbic system. So this is what it is, okay, in the human. Okay. It's not very extremely big in the human. That's one of the things that I was trying to figure out, but I didn't find good resources about it. You know that if you plot the size of the cerebellum compared to the size of the forebrain, in many animals it's like almost complete, static ratio between them. It's very, very similar, the cerebellum. But the hippocampus, if you'll see later, the hippocampus of rodents looks much bigger compared to the brain than in humans. So there are different ways to look into it. One of the things that I wanted to show you, which is, it's kind of, I thought it's important, because of the reason I told you before, that if you look at the hippocampus in the brain, you'll see that, and then the next thing that you always see is these type of slices like that. Okay, so you'll see this type of, people will tell you this is the hippocampus, and then it's kind of difficult to realize what the relation between that thing and that thing. Sorry, is it like when they say CA1 is here and dentate gyrus is here and CA3 is here, because it looks a little bit like that, right? But it's actually completely not like that. It's that if you would slice it this way, the coronal slices, okay? You all with me? Okay. So if you slice it in this way, so it's like as if you have something like that, and it goes like that. So if you cut it this way, you get this dentate gyrus. We'll talk about it in a second. And then the two kind of things that holding against each other. If you would cut it horizontally, you'll get a different. You'll still have the. I don't know if you see that. Is it in focus? For me, what? Not so much, right? Can you? Is there a way to change? Anyway, if you'll cut it horizontally, you will get also all the different parts of the hippocampus that we'll talk about it later, but they will look differently. And lastly, if you cut it sagittally, this is kind of strange because in humans and rodents it's different, but if you cut it sagittally, which means that you cut it this way, okay? Then you get these kind of funny things, like you get the dentate gyrus all around, okay? The dentate gyrus all around here, okay? In a way, how would you say, circled by the CA1 and CA3 areas. So it's kind of, it doesn't matter how you slice it, you always get all the regions, but the classical way is the coronal slices. In rodents it's a little bit different. So this is what I told you in terms of size. You see the size of the hippocampus compared to the human forebrain, and in rodents it's huge compared to the forebrain. And actually in rodents, because the way it actually sits in the brain, you see the way it sits here. So it goes from it goes all the way, kind of hugging the ventricle, if you like. Okay, so then if you cut it sagittally, you get the classical picture of the hippocampus. Okay, so this is what usually a slice of hippocampus would look like, and this is what you will see in almost any synaptic physiology talk. Okay, so this has been seen already very early on by different anatomists. One of them is Laurent Deneau. Oops, sorry. We used a Golgi staining. You have heard of Golgi staining? Everybody knows? What? No? Okay, so Golgi staining is, how would I say? It's probably the most important historical methods of neuroscience. It was developed by Golgi, but it was used mostly by Ramon y Cajal. He developed it to an art, and the idea, it's a silver-based kind of staining. The idea is that you make some solution, and you put your brain slice in it, and the strange thing is that it's being picked up only by a very small percentage of the cells in the brain. So if you would put, okay, let's just very quickly go back. This is what the hippocampus looks like, right? This is like the cells in the hippocampus. Each one of them is showing you a color. Now imagine that all the colors would be in black, okay? So you would just see something that looks something like that, right? There's no way you can actually tell, anything about the anatomy of that thing. Everything is black. So, luckily enough, these things kind of only several cells are picking it up. So you can actually see individual cells with the dendrites and the axon, and can follow them with an optical microscope. And this enabled the first anatomist of the brain to be able to kind of understand the circuits. And this is what they were doing. So Cajal, this was almost everything he was doing. Laurent Deneau also did physiology because he was working later on in the 30s or so. I started with this one because in this one you mostly see the cells, and I also wanted to tell you that this that's something that again I haven't figured out completely, but the CA1 and CA3, and this is shorthand for conuro ammonius. And that's the idea is the first anatomist of the really ancient, I think was the Greeks or something. They thought of that as a horn, okay. And then this is like the ammon horn is like it's a horn of an deer or something like that. I don't know. So this is where this name CA1 and CA3 comes from. So just region of this horn. Except that, again, it doesn't look like with the geometry exactly, so I'm not sure 100% about this explanation. But this is certainly the shorthand for CA1 is shorthand for that. Okay, so what was very impressive about Cajal was that you see these small arrows, okay, which he kind of said, information coming from here, going through these axons, and then travel, and then comes back on these axons. Okay, so going to the dendrite, coming back through these axons. This is, I don't know if, I was assuming that he done talk to you about that last year, but this was a major step in neuroscience, because he called it the rule of dynamic polarization. So that every neuron had a polarization, that information coming from the dendrite through the soma and through the axon. And this was before people actually realized that there are things like action potentials, because they were not recording at the time from these neurons. It was all dead tissue. And the other thing is that they had this huge argument about whether neurons are individual entities. Okay, whether a neuron is a neuron or it's a part of a network, it's a meshing network that everything is connected to everything. And Golgi was actually working on the dentate gyrus, trying to claim that this is what happens, that everything is connected to everything and it's just like, there are blobs and stuff, but it's not individual neurons. Cajal was mostly working on, to prove his point, was working on the cerebellum, but it wasn't until the invention of the electron microscope that people could actually see by eye that neurons are different entities. Okay, so already Cajal kind of figured out the direction of what's going on in the hippocampus, but we know a little bit more today about that, and basically the hippocampus is, as I told you, it's like these two things that holding against each other. One of them is called the dentate gyrus, which is this region. And then the other region is CA3. They're sometimes subdivided into also CA2, then CA1. So each one of these areas is very different from each other. We'll talk about it later. And then there's another area here which is called subiculum. It's kind of part of the hippocampus and not part of the hippocampus. And all that is very close to the cortex, which is going here as a special part of the cortex that we'll talk about it in a second. Okay, so there are three major excitatory neurons in the hippocampus. The dentate gyrus neurons, the dentate gyrus neurons here, the CA3 pyramidal neurons here, and the CA1 pyramidal neurons over here. The input coming from the cortex through what's called the perforant path, going to the dendrites of the dentate gyrus neurons, but also terminates on the apical dendrites of CA3 pyramidal neurons. The axons of the dentate gyrus are going through a bundle of fibers, which is called the Mossi fibers, also to the apical dendrites of these CA3 pyramidal neurons. And the CA3 pyramidal neurons send their axons through a different bundle, which is called the Schaefer collaterals, to the CA1 pyramidal neurons. So in this figure, they don't look so pyramidal, but if you look at Cajal's figure, he was a better animator. You can see that these are actually pyramidal neurons. They're similar in some sense to the cortical pyramidal neurons, and they have apical dendrites. And the CA3 axons going and kind of innervate this area. Okay, this area of the pyramidal neurons. One of the things that is interesting in rats, I now remember that I forgot to show you this, to bring this figure, which is always an argument between hippocampal people and cortical people. What size what size is the apical dendrite? Because in rats, you see the CA1 pyramidal neurons are here, the basal dendrites are up, and the apical dendrites are down. And the cortex is actually sitting here, and the apical dendrites are going up, and the basal dendrites going down. So it's like, it's just, you can see it. So this is the cortex is covering it from here, and the dendrites are pointing upward. So for example, if you would do something like, and people are doing that, I don't know if you heard last year's talk by Yaniv Ziv, who came here. So what people can do today is remove a piece of cortex and put a lens through the cortex, and then do imaging of the CA1 pyramidal neurons in the hippocampus. And the nice thing is that you have the cell bodies just right there for you. You don't have to go like in the cortex. If you want to image layer 5 pyramidal neurons, you have all the apical dendrites from the top to the bottom. And you can't cut them, because if you cut them, you just cut the cortex. So this is very handy. And also when you do electrical recording. So, that's just a side note. Okay, let's just go over that again, because it will come back and back. Again, perforant path coming to dentate gyrus, mossy fibers going to CA3 pyramidal neurons, and CA3 send the axon to CA1, and CA1 is the output layer of the hippocampus. Okay. So one thing that is not mentioned here is that CA3 pyramidal neurons are highly interconnected between themselves. It's probably the most interconnected area in the brain. So if you kind of take two CA3 pyramidal neurons, they're probably very high probability of connection between them. So this is very highly connected. CA1, on the other hand, has no connection between themselves. So they send output, but they don't have any crosstalk between themselves. So this is kind of almost the dream system for computational neuroscientists, right? You have an input layer, you have a recurrent network, hidden layer, and then you have an output layer that just gets the output out, right? It's like as simple as that. It's not as simple as that, but it looks like that. And almost all of you have done like neural network, right? Not one. So you can think of that as a attractor neural network getting an input from some kind of mapping input from input layer, and then a readout. And this is also can talk to you in the sense of I don't know if you heard of all these echo state machines and chaotic networks that people are talking about today. So you have you can also think about it as in the sense of you have a bunch of neurons here which are doing something which is pretty random and chaotic, and you can't actually control it, but you can throw some input into it and just hope the different input will map to different kind of regime of the area that the system will explore, and then you can have a very simple readout of that input, state, and then just read out. So people are actually playing with models like that today, and they're actually extremely powerful as models. Okay, so
how about the dentate gyrus? Are the neurons?
As far as I know, they're not interconnected as well. It's kind of more like a feed-forward network. So they have the dendrites here, and then as far as I know, I might be wrong though. In general, I might be wrong about everything I'm saying. So you have to say.
And are there other types of neurons in the hippocampus?
What do you think? Okay, we'll talk about it in a second. Okay, so these are so the short answer is of course there are plenty of other neurons, almost all of them inhibitory. So all of them are inhibitory. So these are the excitatory neurons, and we'll mention briefly the inhibitory ones. Okay, so sorry. These are the inhibitors. Okay, so this is a course in physiology, so I'm going to talk to you a little bit about the physiology of these neurons. Some of them very briefly, a little bit. One of them, this one, is extremely, studied neuron. So we'll talk about some of the things about it as well. This is somewhat, especially this one, is the first example why the hippocampus is such a great model system. So you take one type of neuron, you study it very, very thoroughly, but almost everything that you find it, you'll later find in different places where it is more difficult to do the experiment. Okay, so you just have the setup to check these things much more easily than in other systems. Okay, so one thing about the hippocampus, before we go deeply into each one of the neurons, is that you can think of it as a single layer network. So you have the dentate gyrus going to CA1, CA3, CA2, yes or no, CA2, and then this goes to the subiculum and to the entorhinal cortex. And there's kind of a flow of information that is going in one direction. Okay, there's also some feedback loop. So the entorhinal cortex is first of all providing the input to the dentate gyrus. The main input to the dentate gyrus comes from the entorhinal cortex. But it also sends some axons to the CA1 pyramidal neurons, and there are other loops inside. You can see that. Can you see that? Maybe here it would be easier to demonstrate, although it's not kind of. Yeah. Okay. So if no. Okay, this is not a good example. Basically, the input that goes to the dentate gyrus from the entorhinal cortex is going through here to this dendrite, but it also follows outside and going outside this line, and this is what this dendritic tips, the tuft of the apical dendrites of the CA1 pyramidal neurons are. So it also gets some direct input to the hippocampus, to the CA1 pyramidal neurons. But it's mainly one layer, and I want to contrast that. This is a horrible slide. I want to contrast that with other systems, okay? So this is the hippocampus, as we said, and you can see that there's one layer of cell bodies that goes there, and the input is kind of the input is kind of ordered sequentially. If you look at other parts of cortex, so kind of in piriform cortex that you probably talked about with Adi when you studied about olfaction. Piriform cortex is an area of the cortex that is responsible for olfaction. Is a kind of an how would you say, early type of cortex. It's what is called three-layered cortex. So you have only 3 layers in this cortex, okay? But then this is already not organized in this way. So you have like layer 5 and layer 2, and the information is going in different direction within the cortex, up and down and inside and outside. This is even more so in the neocortex, where you have 6 layers of neurons. So you have layer 6 and layer 5 and layer 3 and layer 4 and layer 3 and layer 2, yes or no. Sometimes they're kind of lumped together. But then information can go to layer 4 and then go up to layer 2, 3 and then go to layer 5, and it's all within one area. Okay, so it's not like ordered so nicely, and everybody's talking to everybody. It's a whole big mess. So in some ways, and on the other hand, maybe on the other extreme, you have the cerebellum, which it's layered, but it's also organized in a very similar way to the cerebellum, where you have granular cells which are feed-forward input. They get input, but they don't talk to each other, and then they send the parallel fiber to Purkinje cells, and the Purkinje cells send it out. So this is like a 3 stages. You have dendrite gyrus, CA3, CA1. And here you have granular cells, Purkinje cells out. And the Purkinje cells don't talk so much to each other, and the granular cells don't talk so much to each other. So this is like almost a purely feed-forward network. This is a feed-forward network with some reverberations or kind of interconnection. This is a complete mess. Everybody talks to everybody.
So is this related to different stages in evolution?
It's clearly that the neocortex is like, so the limbic system is kind of the most, it's not the oldest system, but it's one of the older systems. And then there was a development of this 3-layer cortex. So in turtles, for example, all you find is 3-layer cortex. You don't find 5 layers or 6-layer cortex. And then in primates, I think in rodents and mammals, you'll find 5-layer cortex. So it does go with evolution, but it's not clear if this is like the earliest. You can debate whether this is like the earliest idea of a cortex that started as a one layer and then wrapped itself into 3 layers and then wrapped itself into 5 layers. You can probably think of that. I don't know if there's any way to make, just hand waving. But it certainly there are kind of, there are things around that, yeah. Okay, so dentate gyros. So these are granular layers. This is very similar to the one that we talked about in the cerebellum as granular layers, but they have a very elaborate dendritic tree. On the other hand, it's a simple one. So like it kind of goes like almost individual dendrites. Okay, it in some ways reminds there is some. It's similar to some kind of early Purkinje cells in electric fish and something like that. Looks similar to that. The inputs is layered in some ways. Okay, so you get some difference in the areas, the different cortical areas are sending the axons to. These are kind of, for electrophysiology, they are considered like a simple neurons. Because, for example, first of all, they have like a very linear membrane. So if you kind of inject current pulses, you get the difference in the current, in the voltage responses are very similar, are linear. Okay, and also they kind of spike. They don't spike too much. It's pretty difficult to get them to spike. Some people believe that this is partially because I will do an analogy to a different granular layer, but it's similar in the sense that these are small neurons and there are tons of them. And someone has calculated once, I think, in the cerebellum, that if each one of the neurons would fire, let's say, once a second, the temperature of the brain will go up in like 10 degrees or something like that, because there's so many of them, and it's an energetic process. So people really believe that one of the reasons is that the very sparsely spiking is because of that. So they're not spiking too much. But on the other hand, if you just record the activity of the output, there's tons of spike coming out because there are so many of them. The other interesting thing to say.
Why they're not spiking so much?
Why is, I don't know if there's a good reason to say why, but the idea is that you have a lot of them.
Yeah,
really.
If they were spiking a lot, then you would have a problem.
Right.
But we don't understand why they're not.
Yeah, so I can give you like. Again, it's a computational course, so I can say that, all right? There are theories. I don't know if I'll have time to go into them here in the hippocampus. Maybe we'll get to them in the cerebellum, and hopefully you'll get to them at some other point in one other course. But the idea is that started with David Marr. Have you heard about Marr theories? So you know that. So the idea is that you try to get your input and throw it into hyperdimensional space. So inputs that will be kind of somehow similar will be mapped to similar places, but it will be much easier to differentiate between them. So when people think about the cerebellum, for example, they think about it as like the input is coming to the granular cells. You know that the granular cells there are as many granular cells in the cerebellum as the number of neurons in the whole brain. So basically, half of the neurons of the brain are cerebellar granular cells, but they only have 4 dendrites each. So they're not kind of very sophisticated neurons. They're tiny neurons, 7 microns the cell body, and then 4 dendrites, and that's it. Okay, so the idea is that each one of them is picking like 4 inputs and map it to something. And then when you look at the whole input-output mapping between the input that comes into that layer and the output that comes out, is like as if you kind of thrown everything to a huge dimensional space. Now these neurons are, we're going completely off topics, but these neurons are sending the axon to the cerebellar layer, Purkinje layer, to the Purkinje layer, and then each one of these neurons are receiving 100,000 inputs. This is like 10 times more than any other neuron in the brain. Okay, and they act as a little bit like a perceptron. They just get tons of input and send an output. So the idea is that you throw it out, and then it's kind of easy to divide it with really simple mechanisms like a perceptron, and you can learn to distinguish different patterns through that mechanism. I mean, this is very early. This is 60s theory, before even synaptic plasticity was demonstrated. But that's kind of the line of people were thinking. And I think there was not like a major conceptual leap since then, except maybe these echo state machines that people are talking about in this context. Okay, so yeah. The other thing that is very interesting about dentate gyrus neurons is that they are the only, I think in humans at least, the only neurons that have neurogenesis in adults. So you've probably heard about that from Adi as well, I'm not sure. But basically we know today that there are progenitors that kind of doing this asymmetric division into new dentate gyrus neurons, and these neurons are integrated in the circuit and become functional. Now, it's obviously kind of tempting to think that this is related to memory and stuff like that. We don't know almost anything about it. It was also being debated for I don't know how many years. People have shown it in the 60s or 70s, and then no one believed them, and then in the 90s people started to look at it again. But nevertheless, it's an interesting phenomenon. It's the only place I know of. I think olfactory neurogenesis works in rodents, but I'm not sure in humans. And people are still looking for cortical adult neurogenesis, but it doesn't look very promising. Okay, CA3 pyramidal neurons. So these are pyramidal neurons again. They have basal dendrite, they have apical dendrite, and they have these mossy fiber boutons, which are kind of huge areas where they get a very strong input from the dentate gyrus neurons. So these were at the beginning were good candidates to study plasticity, but one of the problems is because of this very highly interconnection between these neurons, it's very difficult to work with them, because every spike that is kind of happening in a CA3 pyramidal neurons is kind of immediately echoed by the system, and you get like a lot of activity that you don't control. So this is why this system is not that great for studying this type of thing. Okay, and the king of the hippocampus is CA1 pyramidal neuron, which, as I told you, getting input from the Schaefer collaterals from CA3 pyramidal neurons. For the tip of the dendrite, it gets input from the entorhinal cortex, and it also gets some, and it sends the output to the subiculum and also to the fimbria. We'll talk about it in a second. Okay, what you can also see here is that there's a different set of names that you will hear, which is stratum oriens. Have you ever heard of that? Stratum oriens, stratum radiatum, stratum pyramidalum. So these are the names of the layers that are kind of the input layers and the output layers of the hippocampus. So we said it's a one-layer structure. It's a one-layer structure in terms of cell bodies. Okay, so there's one cell at any given place. There's one excitatory neuron. but there are also all these fibers that are running around, and this is kind of the division into these layers of input. Let me stop for 1 second. We have to. How are we doing with time? Jesus. Okay, let me do like a few more slides and then we'll take a break. Okay, so the electrophysiology of CA1 pyramidal neurons was exhausted to, you can't even imagine how much. So these are the ion channel types that you can find in CA1 pyramidal neurons. Okay, so you have a sodium current, you have persistent sodium current, you have calcium with T-type, N-type, L-type, probably P and Q type as well. You have potassium channels, A, D, K, M, and you also have calcium-dependent potassium channels. What? So this is just a list. So these two are ion channels, or at least currents, that get sodium into the neuron. Okay, these ones are sodium channels that get calcium into the neuron. These are potassium channels that get potassium out of the neuron. Okay, so for example, here this is like a demonstration. What you see here is, imagine you give a neuron a current pulse and you depolarize a membrane. Okay, this is what you see here. You put a current pulse and you depolarize the membrane. Okay, but so.
I was talking about the ADKM.
What about them? What type of current are they? Different type of potassium channels, of potassium current actually, because channels are one thing, and so electrophysiologists usually measure current, and then they kind of describe the current in this way, and then later on you try to find out if there is a specific type of ion channel that is responsible for this current. And usually there is, but sometimes, because the current were found before the channel was, there's a mix-up. So I'll give you an example, like persistent sodium current is a case where you do a voltage step. You take the voltage from, say, -60 to -40, okay? And what you do in voltage clamp, you record a current that is an inward current, is going in, and it stays in all the time, okay? So this, at the same time, there's a current, as it's a sodium current, that it will be transient. It will go, you'll get a current that goes in, but then it will decay and will go to zero. So there are two types of sodium current. Sodium current and persistent sodium current. But no one has ever actually found a persistent sodium channel. A channel that you open, you do a single channel recording, you do a voltage step, and the current just stays there. There is no channel like that. So what it could be the same sodium channel with a different state, because there are probably like many states that they could be in. And the other thing that it could be is that it could be a combination of many sodium channels acting together to give you that result. So this is what some people believe is what's called the window current. So at a certain voltage, there's a probability of the sodium channel to be open and a probability to be closed, and just a population of sodium channel will be kind of changing. Some of them will close, but others will get open, and the total amount will give you like more or less a constant current.
Yeah, but if this is true, shouldn't you in single channel recordings also see channels opening some time after the step, not immediately?
They always do that.
So you see that?
Yeah.
Okay.
Yeah, sure. Okay, so you have all kinds of current reactions different current playing a role, and then people trying to put that into, say, models of say, one pyramidal neuron, reconstructed neuron, and try to see if this is actually describing what they see in experiment. And in the experiment, you can do other things, not only voltage climb. For example, you can look at what a spike look like. So you can inject a current, and then you get a train of spikes. But you can also give a brief current and get a spike, and then these spikes look a little bit different than these ones, because in these ones you kind of hold the current, you hold the voltage up. So different channels of these combinations will kind of come into play. In this case, you only depolarize the membrane a little bit. You get a spike, and then this depolarization of the spike will not activate all these channels, for example. And you get kind of funny things, like you get after depolarization, after the spike. So the spike hyperpolarized, but then depolarized, and then goes down again and hyperpolarized again. So you get after depolarization, after hyperpolarization, and this is a short time scale. If you look at a longer time scale, this thing here would be that thing, and then you have a long after hyperpolarization. So there's very complicated phenomena that happens in these neurons. Some of them are also due to things that happens in the dendrite, because this spike that you saw in the soma, if you record in the dendrite, it actually back-propagates into the dendrite. And then it activates other ion channels on the dendrite, and these ion channels are changing the membrane potential. So it's a very complicated system of many non-linearities that happening due inside these neurons. And I'll give you two examples. One of them is that you see that if you inject a train of action potential in the soma, you have backpropagating action potential in the dendrites, but the amplitude of this backpropagating action potential is going down. And at some point, it just completely fails. So these are not action potentials anymore. They're really tiny things that actually passively decay from these action potentials here. But if you activate, how do you call this fiber that comes into this area? The Schaefer collaterals. If you activate them, then these neurons, these dendrites will be depolarized, and then suddenly these spikes will become much bigger and will be able to invade the apical dendrite. Okay, so some people have the idea that the Schaefer collaterals actually are a gating mechanism for plasticity, if you like, in this area, because you don't get, we'll talk about it later, but you don't get this action potential back into the apical dendrite of the CA1 pyramidal cell unless you activate the Schaefer collaterals at the same time. But if they do, then they depolarize these areas, and then you can actually induce plasticity from the fibers that are coming from the cortex towards these dendrites. Okay, I see that you're getting very tired, so let's take a break here. So one of the interesting phenomena, basically, I'll say very briefly that there was a little bit of a revolution from, I would say, 1994, when people started to be able to do electrophysiology from dendrites. And everything I'm telling you now was done more or less in parallel with other neurons in the cortex, some of them in the cortex and in the cerebellum. But many of the things have been found first. So the backpropagating action potential was first seen in the cortex, but this phenomena of invasion and gating of the tuft was actually found first in CA1 pyramidal neurons. And also the next phenomena that I'll tell you about was kind of co-found at the same time, but was described first by people working on CA1 pyramidal neurons. I put this slide because that was one of my first projects in my PhD, was inspired by this work. and I'm still working on things that are related today. It was a very peculiar phenomena that people started to go and patch the dendrites. In this case, a guy called Dax Hoffman in Dan Johnson's lab went and patched the dendrite along the apical dendrite of this C1 pyramidal neuron and was recording one of these currents, in this case A-type current. So this is a voltage step and you get this kind of current response, potassium current. And peculiarly, it's a with distance, so this is the distance from the soma, he saw that there is more and more and more potassium current in the dendrite. Okay, and this was kind of very mysterious. Why is it like that? What is the function of that? Is it important or not? So we were doing some modeling on that, but at the same time, people were recording a different type of current, a very interesting current called IH. And this IH also did that thing, okay? Also kind of increased with distance from the soma. So it's kind of, it's still a mystery why, what the function of that. We have some ideas, but it's still a mystery. But what is interesting, I haven't put it on the slide, that today we know the molecules that are responsible for that. So you can actually find the molecules that make these channels go to the dendrite and kind of be expressed more there.
The injection is at the same place where the recording is, or is it at the soma?
No. The injection it's what is called it's cell attached mode, basically. So they go and patch. They do single channel. Sometimes, so this is not a single channel. This is a patch where you have a lot of current, but they also do it with single channel, and you just go and patch small patches of the dendrite. And there is kind of variability, and some of the variability is due to the fact that your patch size is not exactly the same every time, and some of it just natural variability, obviously. But again, I don't have a slide for that, but you can also do immunohistochemistry and stain the channels with specific antibodies and see that you have more of them in the dendrite. So there's no argument about the fact that there are in the dendrites more. What the function of that is under debate. Why is it important? Another thing that was shown first in dendrites of C1 pyramidal cell is things of supralinear summation. So this is a very neat technique. What they do here is they have a C1 pyramidal neuron filled, and you have a pipette in the soma, and you have a fluorescent dye in the pipette, so the cell is kind of bright. You can see the cell. This is a 2-photon image of the neuron. But what you can do if you in 2-photon, you can actually see individual spines. We haven't talked about spines, by the way. This is kind of, I took it for granted that you know about spines. But the dendrites of C1 pyramidal neurons are covered with these mushroom-like protrusions that are called spine, and the top of each one of these spines is an excitatory synapse. So basically, if you could, so what they do here is that they use method called 2-photon uncaging. The idea is that you have, these are excitatory synapses, so they are activated by glutamate. So there are special chemical compound which is glutamate that is caged, is not active. It cannot bind to the synapse. But if you shine light on that, it breaks a bond, and then the glutamate is is released and can bind to the synapse. So traditionally, this was done with UV laser, which would break the bond. But today, it's possible to do it also with 2-photon. And the advantage with 2-photon is that 2-photon has a very good spatial resolution. So you have like a ball of about 1 micron where you can release the glutamate. So you can actually, with the 2-photon, you can go very close to this spine and release glutamate. So basically, you put glutamate in the bath. You don't throw the glutamate there. It's too small. You can't see it or anything, right? but you have glutamate in the bus, but you release the glutamate only there. Okay, and and then what you can do is you can kind of select different spines and activate specific synapses one after the other. Okay, there is of course a debate whether this is real kind of similar to what would happen in a real synapse. It's obviously not exactly the same, but it's still similar in the sense that if you put you record the voltage at the soma, it looks like each one of them gives you something which looks like an individual EPSP. Okay, single, and you can actually kind of do what is called minimal stimulation. So you can kind of also have an extracellular electrode here and stimulate an axon until you get only, the minimal response. So if you get a little bit more current, you don't get any response. If you get less, you get more, you get a response. You get less, you inject less current, you don't get any response. So you kind of convince yourself that you're stimulating only one neuron, one axon, and compare this EPSP to this one axon stimulation, and convince yourself that this is pretty similar to a real EPSP that will come from this synapse. And then you play the game of what happens if I activate 1, 2, 3, 4, one after the other. And what you see is that at some point suddenly you get more voltage response than the linear summation. So the linear summation should give you this line, but you get above this line, you get a jump. Okay? You recorded the soma, right? And this is explained by a combination of sodium channels that are voltage dependent. So if you activate several synapses at the same time, the voltage increases, and then you release. You can recruit sodium channels. Okay. And the other thing is NMDA, which is also working in the same way. We'll talk about it later. But you depolarize the membrane, you release the magnesium block, and then NMDA become active. So the glutamate that you release binds also to NMDA receptors, and now you get more current. Okay, this has been later shown as well in cortical pyramidal cells as well. So these are kind of examples of the same thing, but here you get a sodium spike. You see, you get suddenly something very, very sharp, transient in the dendrite. So all in all, the idea is that, just to say, the whole thing that I'm trying to tell you now here is that you remember I told you about this neural network model, that you have an input layer, then you have a recurrent network, and then you have an output layer. But the output layer itself is extremely complicated. These neurons getting different type of inputs, okay, on dendrites, which are highly active and full of all kind of ion channels, and the mapping of these input that they get from here to the output that produces completely non-trivial. Okay, it depends on the temporal aspects of how many spikes, where the spike comes to. Do they arrive on the same branch or not? Do these inputs are gating what happens to these inputs, right? Because if these inputs are coming, then the backpropagating action potential comes to that layer. It's very, very complicated. Okay. So this is the first part of the complication, and then the second part comes with, are these all the neurons that we have in the hippocampus? And the answer is no. We have all these inhibitory neurons. Okay, and what you see here is the following. You have these are the layers of the input. So this is stratum radiatum. This is a pyramidalum. That was a neurons somata line, and radiatum. And each one of these neurons this is where the cell body is. So some of them are inside the same layer of the pyramidal neuron. Some are below, some are above, some are very high up. Okay, and these are the dendrites. So this is where the inhibitory neurons collect their inputs from. So, for example, this neuron collects the input from all the layers. Okay, and this which one? This one, for example, is only interested in what's going in this layer. Okay, we'll see one of them in a second. Actually, you could see that one. I forgot the name of it, but you can saw it here already in Kachal's drawing. I wanted to. Why is it? Oh, sorry. Anyway, it doesn't matter. Yeah, we can look for it later. There's probably a more intelligent way to do that. Sorry. Okay. And then these boxes are telling you where the neuron is sending its output. So this one, for example, collects the input from all the layers, but sends the output only to the layer of the somata of pyramidal neurons. This one is very local. It collects the input here, but it sends the output there. But actually, it also sends the output far away. So it's a complete it's a complete different story of what the inhibitory neurons are doing, and they're certainly not like this kind of what you probably have in mind as something that collects input locally and inhibit locally. I will just tell you a story, a short story that I haven't put in a slide, but there's an interesting paper showing that if you stimulate a CA1 pyramidal neuron, you recruit inhibitory inputs that are then this only single pyramidal neuron. You stimulate it, it recruits inhibitory neurons that goes and inhibits the same dendrites of that neuron. So there are all kinds of complicated loops there that we really don't understand yet.
Together with these inhibitory neurons, why are we considering it still a single layer?
Yeah, we because because we are kind of chauvinist people who are interested in excitatory neurons. I don't know. I mean, people are interested in excitatory neurons.
So it's
obviously
excitatory, but
yes,
if we take
yes,
the other
yes.
into account.
Yes, I mean the cortex as well has like almost, I think, more types of inhibitory neurons than than the hippocampus has. And they're also scattered around in all layers. But if you'll just, if you'll stain the, so you know that, I'm not sure actually in the hippocampus, but in the cortex, it's certain that 20% of the neurons are inhibitory. So if you say if you do what we call the Nissel stain, you just stain the cell bodies, then you see the layers, and the layers are actually marked by the excitatory neurons. So, yeah.
So how many neurons here are inhibitory
Compared to the cortex, you mean? I don't know. In what sense? Like, if is it 20, 80 as well?
No, how many? Yeah, so what percentage of the hippocampus?
I don't remember the number. I would guess it's very similar, but I'm not sure.
So this is only the inhibitory neurons of the CA1? CA3, and we have others?
Again, I don't remember. I think there are different ones, yes, in different areas of the hippocampus. There would probably be similar types, but there could be combination, could be different, because even for the sake of the dentate gyrus neurons have dendrites that are going only in one direction, right? They don't have basal and apical. They have only so-called apical or just dendrites. So they don't have. There's nothing to inhibit on the other side in that sense. So there probably wouldn't be at least axons that would go to inhibit that.
CA3 pyramidal neurons have spines too?
CA3, yes. And dendrite gyrus neurons, I think, almost also. Yeah, they all have spines. Yeah, just some example of these inhibitory neurons. So you can see again, this is like a filled in. So this is what you do here. You patch a neuron, you fill it in with some kind of a dye, in this case biocytin, and then when you fixate the tissue, you can develop it and stain everything in black. So you can see here the dendrites are in one layer, the axon goes to a different layer, okay, and the responses are completely different between these three types. You see that they kind of not too different in terms of like the dendritic arbor, but the axonal ones going to different places. This is, do you know what that is? This thing in the current? This funny thing that, you know, if it would be a passive neuron, you inject a hyperpolarizing current, you should see something like that, right? Like an RC circuit. But this is going back. Any idea? That's the same current that I told you about before. This is called IH. So it's kind of a current that is open when you hyperpolarize the neuron, and it depolarizing you back to a more depolarizing current. And if you depolarize, it closes up, and then it tries to hyperpolarizing you, and basically try to keep the neuron balanced. It kind of doesn't like to get the neuron completely out of control. Okay, all in all, the circuit is something like that. You have the entorhinal cortex talking to the dentate gyrus, talking to CA3, talking to CA1, talking to the subiculum, talking back to the entorhinal cortex in different layers. So this is layer 2 and 3 that are talking to the dentate gyrus, and the output of the hippocampus is going to layer 5 and 6 of the entorhinal cortex. And the entorhinal cortex is kind of communicating with the neocortex. At the same time, you have also output that goes to subcortical regions, such as the amygdala, the limbic system, other places. And there are also these kind of feedback loops that, you know, cortex can send things directly to CA1 and other feedback loops. So this is very schematic, and it doesn't take into account all the inhibition and the local circuits and all that. But it's still, as you see, complicated enough. Just to show you that this is really kind of very simplified. If you look at this single arrow that is going here to subcortical region, this is what it looks like when you look at the, you know, what the hippocampus is talking to different nuclei of the amygdala and how they actually communicate back to the hippocampus. So, you know, if you want to solve the brain, there you go. And this is not all, of course, because this is just the beginning. Did you notice this notch here? This is small notch around here that we kind of completely ignored before. The axons that are going from CA3, they are going through the Schaefer collaterals to CA1, but they're also going outside the hippocampus. Do you know where? What? They're going. What? Exactly, it's going to the hippocampus on the other side. So this is what the hippocampus looks like on one side, and this is the other side. And then this axon going here to these things kind of lie just below the corpus callosum. Okay, and then the axon goes out and then back again into the hippocampus on the other side. And this is called the lyra davidis. You know why? I didn't know as well. It looks like a little bit like a harp, so it's like David's harp. It's like singing. But this is kind of very complicated because, not so much because that I could understand that in the way that CA3 are talking to the other side of the CA3, but apparently it's not only the CA3 that are talking to the CA3. It's also the dentate that is talking to the dentate, and the CA1 talking to the CA1. Now, dentate to dentate, I can understand. CA3 to CA3, I can understand. CA1 to CA1, I cannot understand, because it's like CA1 don't talk to each other, you know, when they are in the same room. Why do they talk to the CA1 on the other side? I don't understand that. But apparently it's true. I don't know how strong the evidence for that are. Just because I can, one of the problems is that, say, if you put a virus here, for example, and then trace it to the other side, you have to work hard to show that it's not picked up by the axons of the Schaefer collaterals, kind of transfect CA3 going to the other side and go to the Schaefer collateral on the other side. So it's kind of tricky, but I think people have done that. This is an example where they did it in Dent and Dryus. You can't actually see it very well anyway. So, but basically it goes, they're talking to each other. So it's very complicated. Okay, so enough of anatomy and single cell physiology. No, we're going now for single cell physiology as well. But few things that make the hippocampus, now that you know how it looks, few things that makes it great system. First of all, it's a single layer and with a very concrete lamination. It's unidirectional mostly, and you have many contacts. So if you kind of pick up an axon, you can kind of it almost doesn't matter which neuron you will record from. You'll find some connection. So we'll talk about it. Maybe I'll go back to it go a few slides back for that in a second. The synapses are very plastic. So if you want to study synaptic plasticity, it's a great place to work. You can take neurons and grow them in cultures. So you can actually work on individual neurons and kind of on connection between two neurons. And I'll show you an example of it just before the end of that talk, if I'll ever get to it. You can kind of make slices that will hold on for a long time. Just I want to give you like one brief introduction to that, because I don't know if I'll put a slide on that before. So imagine that you patch a layer 5, CA1 pyramidal neurons here, and you want to study plasticity of the synapses. You patch a neuron here, and then you can stimulate these axons. You just put a stimulating electrode here, and you stimulate an axon, and you can kind of you're almost sure that you'll have a synapse, right? And you can kind of put one stimulating electrode here and one here, and then this one will give you and you can kind of do a plasticity protocol with this one, and at the same time doing a control protocol on the same neuron with exactly the same conditions on a different fiber that you don't induce plasticity. So it's a perfect system. To get that in cortex, forget it. There's no way. So this is why it became so popular. And indeed, some of the most important discoveries were done in the hippocampus. For example, the fact that about this thing that in EM you have symmetric and asymmetric synapses. The symmetric synapses are the inhibitory one, and the asymmetric are the excitatory one. And this has been shown first in the hippocampus. The obviously long-term potentiation and depression, and short-term and whatever term of potentiation and depression that you would like was found in the hippocampus. There's a lot of issues about oscillations and rhythms. We'll try to talk about it next week on Thursday. And obviously things that has to do with epilepsy. Okay, synaptic plasticity. So, how many of you have ever read this paper, by the way? None. Okay, so that's a seminal paper by Tim Bliss and Lomo. They were both of them working in the lab of Per Anderson. It has been cited more than 4,000 times, and it's like probably one of the most influential papers ever in neuroscience. And no one knows it for the good or bad reasons. So 73. This is kind of in a way ingenious paper in some sense, at least in technical one. So this is in vivo in a rabbit. Okay, it's not in slice. And what they did is they took the two sides of the hippocampus. One side is a control, and one side is an experimental side. They put a stimulating electrode on the perforant path, the electrode that goes, the axon that comes from the entorhinal cortex and going to the dentate gyrus, and then they record in the dentate gyrus. This is the extracellular recording, it's field potential. Okay, and they do it in both sides, so they can stimulate in one and record in one, stimulate in the other one, record in the other one. Is there anything else to show here? No. Okay, so this experiment, pretty simple. So you give the stimulation and you call the field potential, and you kind of establish that every time you give a stimulation you get some. This is what they call the population EPSP. So they record here, okay, in this area, and they kind of get. So this is where the axons of the entorhinal cortex are synapsing on the dendrites of the dentate gyrus neurons, and they record the population activity, the voltage due to many of the synapses are kind of being opened at that time, okay? And this is what you get every time you give a stimulation. You get some voltage, say 2 millivolts. Then you give a tetanic stimulation, so you give a train of strong impulses to the perforant path axons. You get a transient increase in the population activity, a reduction, and then after it stabilizes, you get stronger response that you had before. Okay. This is we can ignore the rest here. It's important, but it's not that important for our purposes. So basically, by activating the presynaptic axon, you cause increase in the response of the population. This is today it sounds almost trivial, but at that time was the first time this was shown, and obviously come over a history of thinking of what's the main plastic mechanism in the brain should be in synapses between neurons. So this was the first demonstration of that, and you can actually do it several times, one after the other, and kind of each time get increase in the potentiation. Later it has been shown that you can also get depression due to protocols. We'll talk a little bit about it in a few minutes. So we know today that this is actually, I'm not sure why I put this slide here. Okay, maybe we'll come back to this slide later, but just I wanted to kind of put, you all know that today that there are AMPA receptors and NMDA receptors. Back then they didn't know that. And they also didn't do it like very cleanly, so they couldn't actually figure that out. But so few other phenomena. One of them is that you get almost always short-term plasticity, but not always. But the long-term plasticity is kind of is saturated. So, for example, if you give a tetanus stimulation, you get almost nothing. Then you get potentiation for a short term, but then it saturates, it decays and stabilizes on different value. You do it again, you get another one. You see that the baseline all the time increases. At some point it saturates. You don't get more potentiation, long-term potentiation, but every time you give the tetanus, you get this transient. Okay, so this is one thing. The other thing is too, which is people have shown that you need protein synthesis in order to get the long-term part of that. Okay, so for example, if you block it with an isomycin, protein synthesis, which is kind of I know this is not kind of the nicest way to show it, but you basically lose the long-term part. Okay, so this is what you would get, and this is with azithromycin. You get basically back to baseline. So transient increase, but back to baseline. I want to, at that point, give you a warning that there are, I don't know how many papers dealing with LTPs. Okay, but they tend to show you almost always things like that. Okay, so it's like, so what that means is that they kind of, during that time between 3.5 to 4.5 minutes, they gave like, I don't know, 10 stimulations, and they average them up, and then you get this average plus minus whatever standard deviation. Often when you happen to see the raw data, and then on top of that, they kind of present the data in terms of percentage, so you kind of percent change from baseline. So often when you look at the data, the raw kind of points and traces, it looks like a whole mess. It's all over the place. So like you have like a cloud of responses, and then the cloud goes a little bit up. But the thing is a mess. So if you kind of ever want to make the relationship between a perceptual learning rule and an LTP, you have to know that it's a completely different kind of world, in the sense of the amount of variability that you have in that system. It's extremely noisy, and it's very sensitive to all kinds of to things, the experimental cases, and this is also the reason maybe why there are so many works about that, because kind of you change one thing in the internal solution of your postsynaptic cell, and your whole kind of result is changing. So it's a very tricky thing. It's obviously there. I don't say it's not, but it's very, very complicated to completely characterize it. This is what I wanted to tell you before, the things that you can kind of use one stimulus and another stimulus as a control. So this is very nice. The thing that people liked about it, and this is also why it became popular, because it seems to be Hebbian in the sense that it depends on the postsynaptic cell and the presynaptic cell. Okay, so one example for that is that if you do the protocol of LTP, but this time you actually record from the soma, so you have an electrode in the soma, and you depolarize the membrane during the protocol of the tetanus stimulation of the presynaptic fiber, and you don't say in the other protocol, then in one of them you wouldn't see any change in the synaptic efficacy or EPSP, and in the one that when you depolarize during the protocol, you get increase in the EPSP. So this means that it depends on the pre and postsynaptic cell, and these guys, Binnenstock, Cooper, and Monroe, back in '82 already formulated this learning rule, which became very famous. It's called the BCM rule, and the idea is that the change in the synaptic weight is a function of the postsynaptic spiking frequency, the postsynaptic average firing frequency, and so this is a function multiplied by the presynaptic frequency, and this is kind of the rule. And the idea is that at some domain you would get LTD, and in some domains you'll get LTP. So the same synapse can go under LTP and LTD, and it may be depending on the postsynaptic firing frequency. Okay. Okay. So I don't know if we kind of have to stop. So maybe we'll go briefly over the mechanisms at the beginning of the next lesson. I'll just skip to the last slide maybe. Tell you again what we're going to. Oops. Yeah. So on Thursday we'll talk about. We'll finish up with LTP and plasticity, and then we'll talk a little bit about memory. I wouldn't get too much into it because I think it's covered much better in the cognitive course, but we'll just mention it briefly. And we'll talk about spatial navigation, place cells, grid cells, maybe a little bit. Sorry. What do you mean? Sorry.
Which course were you referring to?
You have a course in cognitive science, no? Like psychology, psychology. No? There is one course, right? That two now? Leon. Okay, let's just say that we'll talk a little bit about it briefly. It's and the reason is that it's not so much physiology. It's in the hippocampus, but it's mainly memory. So we'll mention a few things about it. I'm not going to go very deep into that. We'll talk about spatial navigation. We'll talk a little bit about rhythms, and if I have time, I'll also cover some models. Again, this is also not part of that course, so I'll leave it to the theoreticians. Okay. If there is something specific that you want to cover, let me know. And regarding the lectures, I will try today or if not today, so certainly tomorrow to put the list of who is presenting a paper when. And the first papers are already sent to the people who are going to present it. Okay.